Sample chamber-based light sheet fluorescence microscopes are designed for high content rather than high throughput. Live imaging assays must therefore be performed sequentially and are thus affected by ambient variance.
Our cobweb holder allows stacking and simultaneous imaging of multiple embryos, eliminating ambient variance and increasing throughput.
Mounting embryos in the cobweb holder requires a certain finesse. An explanatory video is ideal for illustrating the sequence of the many short consecutive gestures.
For calibration of the sample chamber-based light sheet fluorescence microscope, first, reliquefy an agarose aliquot in a dry block heater mixer at 80 degrees Celsius. Allow the melted agarose to cool to 35 degrees Celsius and transfer 50 microliters of the agarose to a 1.5 milliliter reaction tube. Mix 0.5 microliters of fluorescent microsphere solution with the agarose at 1,400 revolutions per minute for 1 minute, and fill the slotted hole of the cobweb holder with 10 microliters of the agarose fluorescent microsphere solution mixture. Aspirate as much agarose as possible until only a thin agarose film remains, and allow the agarose to solidify for 30 to 60 seconds. Fill the sample chamber with autoclaved tap water, and slowly insert the cobweb holder into the sample chamber. Move the slotted hole in front of the detection lens and rotate the holder to a 45-degree position relative to the illumination and detection axes. The cobweb holder should not be visible in the transmission light channel. Switch to the appropriate fluorescence channel and adjust the laser power and exposure time so that the fluorescent microspheres provide a proper signal. Specify